Arthropods are the most successful animal phylum. What makes them so successful? Partially, it may be their mechanism of gas exchange. In a broad context, all eukaryotic organisms require a mechanism of gas exchange. There must be a way to acquire oxygen for cellular respiration and then expel the CO2 that accrues as a byproduct. Gas exchange in insects is unique in the fact that respiration is not coupled with circulation. Insects utilize various patterns of gas exchange, one of which is discontinuous gas exchange, or DGE. This process has independently evolved more than 7 times in the Arthropoda phylum, 5 of those times within the Insecta subphylum. DGE is obviously evolutionary advantageous, but how? Can this help us understand the evolutionary success of arthropods? Gas exchange occurs through the tracheae, a branching network of bronchial tubes. The tracheal system has openings, known as spiracles, that connect to the outside environment and act as channels for gas exchange that are gated by spirochelar valves. DGE is a respiratory pattern where spirochelar valves open and close in a predictable pattern in order to get oxygen into the tracheal system and CO2 out. DGE in insects is largely composed of 3 phases. The length of each stage and the frequency of these stages vary from insect to insect. However, in a broad context, the stages of discontinuous gas exchange are the close spiracle phase, where oxygen in the trachea is consumed by cellular respiration and CO2 is produced as a byproduct. The partial pressure of oxygen begins falling, and once a threshold pressure of 5 kilopascals is reached, a signal from the central nervous system causes spirochelar valves to flutter and enter the next phase, where the fluttering of spirochelar valves causes oxygen to move down the pressure gradient from outside of the insect's body into the insect's body. Finally, in the last phase, while the valves are fluttering, the partial pressure of CO2 continues to build up inside the insect's body, finally reaching about 4 to 6 kilopascals. This prompts the complete opening of the spirochelar valves and for the CO2 to exit the tracheal system.
In addition to DGE, insects may utilize other gas exchange patterns. Cyclic gas exchange is similar to DGE in that there is rhythmic repetition of CO2 release. However, the spiracles never completely close, so the rate of CO2 release is never zero. Continuous gas exchange is characterized by spiracles constantly in the open phase, allowing for a constant rate of gas diffusion. Various hypotheses have been proposed to explain the evolutionary advantages of DGE. The hydric hypothesis states that DGE reduces respiratory water loss. This is the oldest hypothesis and has the most supporting data, but has the most dissent as well. The chthonic hypothesis states that DGE facilitates gas exchange during low O2 partial pressures, high CO2 partial pressures, or both. DGE allows for the generation of greater partial pressure gradients between the animal and its environment, allowing for more efficient gas exchange. These 2 hypotheses can be combined into the chthonic hydric hypothesis, which states that DGE is a way to facilitate gas exchange without excessive water loss. Finally, the hypothesis advanced by a primary article is the strolling arthropod hypothesis, which states that DGE minimizes the risk of infestation of the tracheal system by parasites such as mites, bacteria, and fungal spores. When arthropods are infested by these external parasites, there is a high risk that the parasites will utilize open spiracles to enter the respiratory system. Therefore, arthropods with external parasites are more likely to keep their spiracles completely closed for brief periods, preventing the entry of parasites into their respiratory system.
The White study compared DGE duration between beetles with external mites and beetles without external mites. In order to do so, beetles of 15 different species in the family Carabidae were collected and kept in uniform conditions. Their metabolic and gas exchange rates were measured, and this data was used to determine the gas exchange pattern used by the beetle throughout the observation period. After the respiratory measurements, the beetles were euthanized and examined under a stereo microscope to see if they had mites. The prevalence of each gas exchange pattern was compared between beetles with mites and beetles without mites. When the data was examined, beetles used DGE more frequently when harboring external mites. In non-infested beetles, DGE decreased if oxygen demand decreased, but the same trend was not observed in infested beetles. In beetles infested with mites, there was no relationship between gas exchange pattern and metabolic rate. This suggested that the infested mites weren't using DGE simply because their metabolic needs decreased due to poor health. The image shows the 3 patterns of gas exchange in insects: DGE, cyclic, and continuous gas exchange. This data was compiled using open flow respirometry, and it shows CO2 emissions as a function of time. While both DGE and cyclic gas exchange undergo repeating periods of rising and falling CO2 emissions, periods of zero CO2 emission only occur in DGE. In continuous gas exchange, there is no repeating pattern, and levels of gas exchange are relatively constant. This graph shows a comparison between the prevalence of each gas exchange pattern in infested and non-infested beetles. Far more infested beetles engaged in DGE than non-infested beetles, and far fewer infested beetles engaged in continuous gas exchange than non-infested beetles.
DGE serves as an example of how efficient and evolved the tracheal system of arthropods is. We have discussed many different mechanisms of exchanging gas between organisms and their environment, particularly the opening and closing of stomata in plant species. This type of gas exchange is very similar to spirochelar valves in insects and some other arthropods. In plants, there is an evolutionary trade-off between evapotranspiration and photosynthesis. This is analogous to the trade-off between gas exchange and water loss in arthropods, as predicted by the Clonic hypothesis. In other organisms, such as mammals, the gas exchange process is paired with the circulatory system. By not coupling these processes, arthropods are capable of maintaining high metabolic rates while exchanging gas to keep body weight low to fly. Even though there has been a lot of research in this area, there are also many details that cause dissension within the scientific community. For one, the DGE cycles can differ from species to species, and researchers are actively trying to determine the exact mechanisms that govern DGE in different species and why this variation exists. While a range of hypotheses exist for DGE's emergence, White suggests that they may not be mutually exclusive, as DGE may confer several evolutionary benefits. Furthermore, DGE may be an example of an adaptation, or a trait that provides additional adaptive functions outside of its original adaptive purpose. Additionally, in terms of an evolutionary standpoint, DGE has evolved 7 separate times in the arthropod family, and some species have actually lost the ability to conduct DGE. This can be an interesting opportunity to investigate the link between the selective pressures in an organism's environment and how a species will change over time in order to adapt.